Hi there, welcome to Core Practical 2, which is the vitamin C one. So we're going to go through how this experiment works, how you do it, what you find out, etc. So first of all, our variables, independent, dependent, control. So in this one, the independent is the food type. So most of the time you probably measure different types of fruit juice, but it's your different food type. So type of fruit juice, for example. The second one is your dependent. So what are you going to measure? Um, in a really kind of specific way, what you're going to measure is the number of drops of juice needed to decolorize DC hip. Okay. Your control variables, the things that you're going to keep the same in this one, you need to keep the same concentration and the same volume of DC pip. Then you're going to go into your method. How do you do this experiment? So, those up there. This has a new box. Method. So first of all, you need to do your calibration curve. So your calibration curve is, if you remember, it's a graph. So you need to do first part of the experiment is in order to make this graph. So it's this sort of shape with your ascorbic acid down here. Okay, and the number of drops here. So, in order to generate this graph with exact numbers, etc., that you can use for reference in the later part of your experiment, this is what you need to do. So, find a box to make the calibration curve. So first of all, you need to make up the vitamin C solutions. Now, another word for vitamin C is ascorbic acid. Okay, so make up the vitamin C or ascorbic acid solutions. They're going to be 5 different concentrations. Remember, we would use 5 different ones for our independent variable, and we need to use percentages. Okay, so just make it up if you don't know. So do like 0%, 1%, 2%, 3%, 4%. So I got 5 different values and I've given exact numbers. Alright. Next, I need to measure out a known concentration of DC pip. Into a test tube. 5 ml would be good. So again, use values in your method. Measure, so 5 ml. Rather than mil, centimetre cubed is better, but I can't do cubes on my program here. Next, you're going to add one of your vitamin C solutions to this. So look at how we're not even using the juice in this section. This is just like the setup section. Okay, add 1 vitamin C solution. So that's one of your percentages from step 1 to the DC pip, drop by drop. You gently shake the test tube for a few seconds after each drop just to make sure it's fully dissolved. And we're looking for when it changes colour. Okay, so the DC pip's going to start blue and it's going to change to colourless. So add one vitamin C solution to the DC pip drop by drop until it changes from blue to colourless. Okay, now this is called titrating, adding a little bit at a time until it changes colour. So we could say titrate rather than add. Titrate one vitamin C solution with the DC pip drop by drop until it changes colour from blue to colourless. So we need to know exactly how much we've added. So we would have put it in a burette, which is what we use for titrations, because then it measures how much we've added. Then up the side here, we'd be able to put volume. Alternatively, you can add drop by drop from a pipette, and then up this axis here, you can put number of drops. It doesn't really matter either way. And then you're going to do that for each concentration. So when you've done it for the first concentration, so this axis down here, where I've put ascorbic acid, I should have put concentration. So here we've just done ours in percentages. So, where is it? One. So if you did it with the 0% solution, then it never would have changed colour. If you did it with the 1% solution, then you'll have plotted your X there. Then you'll have done it with your 2% solution, plotted your X there. Then with your 3, plotted it there. Then with your 4, plotted it there. Then with your 5, plotted it there. And then from that, you'll have been able to draw your calibration curve. So this here is called a calibration curve. Okay, so that's how you make your calibration curve. Sometimes you might see it called a standard curve. So you would then repeat for the other concentrations and draw calibration curve. Okay, so that's kind of your setup step, if you like. And you've got another part to the method. So you now need to test your fruit juices to see how much ascorbic acid or vitamin C they've got in. So what you're going to do is you're going to see how many drops do I need for apple juice? How many drops do I need for orange juice? How many drops do I need for pineapple juice? So if, for example, we put some numbers on here, if you tested it out with apple juice, and you needed 6 drops, you'd go across and down and see what percentage of ascorbic acid it has in. If for pineapple juice you needed 5 drops, you go across, down, and you'd be able to work out the percentage. If for orange juice you actually only got 1, you could go across and down and see how percentage of vitamin C it's got in. So remember, ascorbic acid is another name for vitamin C. All right. So the next step on here is to test your fruit juices. So testing the fruit juices. So your fruit juices are your actual independent variable, aren't they? That's what we're actually trying to find out. So first of all, measure the known concentration of DC pip into a test tube. Remember quantity, I'm going to say 5 ml. And next one, going to add your fruit juice drop by drop this time. You need to know how many drops it takes to change colour. So add fruit juice drop by drop until it changes from blue to colourless. Again, remember that's titrating, so you could say titrate the fruit juice into the DC pip. Next, number 3, make sure you shake it in between each drop. Open up here. You shake in each drop to make sure it's fully absorbed, fully dissolved, sorry. Make sure now that you repeat. This will allow you to identify anomalies. Calculating. Don't include your anomalies, obviously, when you calculate your mean. That makes it more reliable. And then you need to look up your results on your calibration curve. So look up the results on the calibration curve. Read along, or across, I should say, from the y-axis to the line and down to find the vitamin C concentration for each fruit juice. Nice and easy. Okay. There is a bit of a problem with this experiment in that lots of the fruit juices can be thick or they can be really coloured and it's hard to see the colour change. It's always useful when you're doing your experiment to have some fruit juice that hasn't got the DC pip with it just on the side, just so you can compare the colour change. So when the DC pip goes clear, you're not going to see a clear solution. You're going to see the original colour of the apple juice. So if you've got just some plain apple juice at the side to compare when it's finished changing colour, it's really, really useful. Again, when you're doing orange juice, it's not going to go clear. It's not going to go blue to clear. It's going to go blue to orange. Okay, so just be aware of that. If you were doing, I don't know, cranberry juice, it's not going to go blue to clear. It's going to go blue to red. So then you're going to have to write your conclusion. What is this experiment going to tell us? What are we going to find out? So you can find out which vitamin C has the most, which has the least. First of all, which fruit juice contains the most and the least vitamin C. And it's always useful in a conclusion to be able to interpret what our results mean. So, what is vitamin C? Now, let's do a little bit about that. So, vitamin C is an antioxidant. Useful to have in our diets. You also get lots of vitamin C in skincare now as well. And what antioxidants do is they neutralize free radicals. We don't really do anything about free radicals in the biology course, so it's a bit of a just learn it. But what free radicals do is they can damage cells, so it reduces the amount of cell damage that happens. Neutralize free radicals to reduce cellular damage. We also need to know how the DC pip changes colour. So a little bit of chemistry as to what happens there. So vitamin C is also a reducing agent. So it reduces things. So remember, oil rig, oxidation is loss of electrons, reduction is gain of electrons. It's to do with that. So vitamin C is a reducing agent. So it reduces other things. So oil rig, let's just write that up in the corner. Those of us that don't have it on the tip of our tongues. So oxidation is loss of electrons. Reduction is gain of electrons. Okay, oxidation is loss of electrons. Reduction is gain of electrons. So if vitamin C is a reducing agent, that means it reduces other things. That means it causes other things to gain electrons. So it gives its electrons to the other chemicals. Okay, so it gives its electrons to other chemicals to use. Look at your oil rig at the top to help you understand that. So DC pip is what we call a redox dye. DC pip is a redox dye. Now, redox is reduction oxidation reaction. Now, oxidation and reduction always happen at the same time, because if something's going to gain electrons, they must have come from something else. So the thing that gives them away is being oxidized, and the thing that's gaining them is being reduced. So you always get redox reactions. You never just get oxidation or reduction. One thing's being oxidized, one thing's being reduced at the same time. So DC pip is a redox dye, which means they change colour when they're reduced. So it changes colour when it is reduced. Okay, when it's reduced, look at your oil rig. That means when it gains electrons. And so we know that vitamin C is a reducing agent. So the vitamin C will give its electrons to the DC pip, and it will change from blue to colourless. Okay. That's basically it, actually, in terms of the science of it. So what I just want to do is just kind of draw it for those of you that prefer things to be drawn. So if this here is our vitamin C, and this over here is our DC pip, what happens is that the vitamin C gives an electron to the DC pip. So you can see the DC pip is gaining an electron, so it's being reduced. The vitamin C is losing an electron, so it's being oxidized. And because we've got oxidation and reduction together, we call it a redox reaction, because they're both happening at the same time. And when this DC pip gets reduced, it changes colour, and that's why we saw the colour change in our experiment. Right, the final thing is our evaluation. Okay, so the last part is your evaluation. I've just pre-done the headings because they're always going to be the same. They're likely to ask you one very specific question, including one of these words. So the first one's reliability. So in this one, when we tested our fruit juices, we tested each fruit juice 3 times. Okay, we can then identify any anomalies. If we identify an anomaly along the way, we should then do another repeat. Okay, and then we take a mean of our results. Don't include your anomaly when you do your mean. All right, so it allows you to identify the anomalies. Then, if you've got time, you can do a 4th one or a 5th one if needed, until we've got 3 results that look right, and then you can calculate your mean. And remember, reliable begins with an R, repeats begins with an R. So reliability is all about doing repeats. Next one is reproducibility. This is a different experiment with the same results as yours. Gives the same results. So if someone else did a different experiment, they also found that orange juice had the most vitamin C, for example, then that's increasing the reproducibility of your experiment, because you can reproduce it. But if you just did the same experiment again, that's just a repeat. But if someone else, or if a different experiment shows the same result, that's reproducible. Validity is only change the independent variable. So in this, the only thing we're changing is a concentration of the vitamin C. Okay, don't change anything else. All other variables must be kept the same. Really, really important. Then it answers the original question, which is maybe which fruit juice has the highest concentration of vitamin C. Oh, sorry, I only changed one variable. We didn't change the concentration of vitamin C. Our variable was the type of fruit juice. Yes. It mixes you up, doesn't it? Because You change the concentration when you're doing your standard curve at the start, your calibration curve. So don't make the same mistake as me. Next one is precision. If something is precise, when you do your results, they're very similar. If your results give repeats that are really similar, the results are precise. If when you repeat it, you get results that are different, then it's not very precise. Accurate means close to the true value. So if a scientist that was really well practiced and worked in vitamin C concentrations every day of their life and was a real expert with the best equipment in the world, what would they get as the vitamin C concentration of that orange juice? Would they get the same result as you? In which case your result's accurate. If they get a different result, then your result's not very accurate. But how can we tell that? We could compare to kind of published results online that someone had got using, excellent technique and excellent equipment. Alternatively, we could draw results onto a graph and compare them, but that doesn't really apply to this experiment. So are our results close to the true value? The next one is the uncertainty of our results. That's to do with the sensitivity of our measuring apparatus. So this time, really, that's due to how large our drops are. So it's to do with our pipettes. Okay, so that's to do with the size of the drops from the pipette, because our measurement was number of drops. Now, alternatively, if we'd used a burette and we'd measured the actual volume that we were adding, it would be half the smallest measurement on the burette. Okay, but actually we just used a pipette drop, so it's the size of the drops from the pipettes. We can't really measure that. The last one that isn't on here is percentage error. And your percentage error is your uncertainty. So it's basically a calculation. Your uncertainty divided by the reading. So you can work it out for each reading times 100. So say we had used a burette, then we could have worked out the uncertainty. It would have been half the smallest measurement on the burette. Okay, and then we could have worked out our percentage error because it would have been half of the smallest measurement on the burette divided by the reading, whatever the volume was that we had to add, make it change colour, times 100, and we could calculate the percentage error. Okay, and that's it.